Hi everyone! So today we're gonna talk about the genetic code and its nature, background, and how did scientists come up with the triplet code that we are using in the central dogma of molecular genetics. So start. By 2001, the DNA sequence in the genomes of more than 20 different species have already been sequenced. So that includes the bacteria, Escherichia coli, the yeast, Saccharomyces cerevisiae, the fruit fly, the Drosophila melanogaster, the nematode, Caenorhabditis elegans, the plant, Arabidopsis thaliana, and humans, or Homo sapiens, had already been deciphered. So these are the following species, or different species, by 2001 that has completed and deciphered their genomes. Later in this chapter, you will see on how the studies of your Caenorhabditis elegans, or the nematode worm, will illustrate the insights possible from the complete sequence of your genetic or genomic DNA. This picture on the left is a tiny roundworm that lives in the soil all throughout the world. And this is also an ideal subject for genetic analysis. And your different micrographs, okay, this on the left will show you several adult worms. The central dogma of molecular biology, or shall we say the gene expression, will tell us, okay, the process by which cells are converting the DNA sequence information to RNA and then decode your RNA information to the amino acid sequence of your polypeptide. And interestingly, Caenorhabditis elegans, because of their almost 20,000 genes, roughly 15% of their genes will encode the same expression, the same manner by this gene expression is performing or showcasing. Okay, so gene expression is technically the flow of genetic information coming from your double-stranded DNA via RNA until it will go into a protein. Okay, so just an overview. In the future presentations or videos, I will be talking more on the process of transcription and translation as how it interplays or how it intervenes the sequence of your gene expression. So in transcription, your enzyme RNA polymerase copies the DNA, okay, and they will produce an RNA transcript. Okay, in translation, this cell machinery will use the instructions in your mRNA to create or synthesize what we call as your polypeptide, or series of amino acids, and they follow the rules of your genetic code. So in this presentation, we also take note the works of Francis Crick, okay, and we also will describe the cellular mechanisms that carry out gene expression. So it may be as intricate as it may be appearing, okay, but the general scheme of your gene expression is very elegant and straightforward. Okay, within each cell, okay, the genetic information comes from your DNA and then your RNA to your protein. Okay, this statement was also set forward by our molecular biologist on the left. You'll see Francis Crick. He explained the central dogma of molecular biology and telling us that once information has passed into protein, it cannot get out again. Okay, so this is Francis Crick. And the central dogma of molecular biology will not explain the behavior of all genes, as Francis Crick himself realized that a very large subset of genes, okay, is transcribed into RNAs that are never, okay, translated into proteins. There are some genes, or there are a large subset of genes that have been transcribed or that have been placed into a single-stranded mRNAs, and they will remain mRNAs. Okay, they will not be translated or never be translated to proteins. And these genes that are encoding the rRNAs and tRNAs will also belong to this group. So In addition, scientists have also later found the certain viruses that contains an enzyme, okay, and that will reverse, instead of DNA RNA to protein, okay, it will be doing or copying RNA to DNA, and that process is what we call as your reverse transcription. And there are some processes in biology that uses reverse transcriptase, okay, in this mechanism. Okay, for example, your HIV or the okay, you have this photo of a scanning electron micrograph of your HIV-1 in green, the most common type of HIV, okay, budding from a cultured lymphocyte. So there are some multiple round bumps that you can see on the surface of the cell, and they will represent sites of assembly and budding of your virion. So let's talk about what reverse transcriptase is. So the reverse transcriptase is an enzyme of a human immunodeficiency virus, and this is a multifunctional enzyme which is responsible for the conversion of your single-stranded viral genome into a double-stranded DNA that is integrated into the host genome by the viral enzyme integrase. So you will see on the image on the left, which is a surface representation of the crystal structure of the wild-type HIV-1 reverse transcriptase based on your PDB3KLF. HIV-1, as I mentioned earlier, is the most common type of your HIV. Okay, it attacks your body, your immune system, your own immune system, and they destroy your CD4 cells. So these cells help your body to fight infection. HIV-1 also can severely damage your immune system and will lead to your acquired immune deficiency syndrome or AIDS. So you'll see on the photo this polymerase and the nuclease, the P66 and the P51. So what are the four general themes of your gene expression? So we have the first, the pairing of complementary bases is key to the transfer of information from DNA to RNA and from RNA to protein. So it is very important that we'll know that the base pairs of the nitrogenous bases, okay, and their pairing and the sequence of how they are being arranged is the central idea, okay, of your gene expression. Next will be the polarities or the directionality of your DNA, RNA, and polypeptides that will help us guide the mechanisms of gene expressions, the direction of where they're going to. Like the DNA in third, like DNA replication and recombination, gene expression requires a very high input of energy and participation of such specific proteins. There are proteins that are participating, and also some of macromolecular assemblies, okay, such as the cell organelles, we call them ribosomes. And lastly, mutations that change genetic information or obstruct the flow of its expression can have a very dramatic effects on the phenotype. So let's talk about, so this is the genetic code, and you will see a picture on the right, the Drosophila wing cells, fruit fly wing cells. So this human CBFA2 T3 GLIS2 fusion protein is a key driver of what we call as your pediatric acute megakardioblastic leukemia, and it confers a poor prognosis. And researchers have found, okay, you will see that a way to express your CBFA2 T3 GLIS2 red, okay, this one is shown in red, in your larval Drosophila wing disc cells, is confirming a major role for your BMP signaling pathway. So the pathway may provide a target for new therapies, and you'll see this picture on your right, nuclei which is shown in green and actin filaments which are shown in purple. So this disease, pediatric acute megakardioblastic leukemia, is a very rare, okay, subtype of acute myeloid leukemia. Okay, that is also can be divided into different subgroups. The first one is your Down syndrome related AMKL, which has a generally have favorable prognosis, and your second will be your non-Down syndrome related AKML, which has sometimes a poorer outcome. So let's take a look at it on how this genetic code is very important in our understanding of your gene expression. So the triplet codons of nucleotides will represent individual amino acids. So we call them the nucleotide triplet as a codon. So each codon is designated by its 3 nucleotides, and they specify a certain amino acid. So for example, GAA is a codon for your glutamic acid, and GUU is a codon for valine. Because the code comes into play only during the part of translation, which is a part of gene expression, that is during the decoding of your mRNA to polypeptide. So in this process, geneticists usually present the code in the RNA dialect or the dialect of mRNA, and it could include your base pairs, the A, G, C, U, as depicted on the next slide. We will be learning on how to read the frames of your genetic code or the triplet codons. So when we're speaking of genes, we can substitute the thymine for uracil to show the same code in your DNA dialect. So you will be learning that if you will be doing reverse transcription in the late. The photo on your right will represent the 61 codons that will represent the 20 amino acids. Okay, and there are 3 codons will signify or will indicate the termination codons or the stop codons. So how do we read the code? Because find the first, okay, the first letter, which is found in the left column, this yellow. Okay, the second letter is found on the top, and the third letter will be found on the right column. So this represents, okay, the reading that is coming from a 5' to 3' direction along your mRNA. So the start codon is AUG, which stands for methionine, and these are the UAG, UA, UGA, which are the termination codons. So geneticists reasoned out the theoretical grounds that codons are composed of only 3 nucleotides, and they will provide the simplest mechanism by which genes are gonna encode the 20 amino acids which are commonly found in proteins. And the gene's nucleotide sequence is very collinear with the amino acid sequence of your encoded polypeptide. Going back, okay, DNA is a linear molecule with base pairs following one another down your intertwined chain. So they can be read on a very linear fashion. And proteins, by contrast, have very complicated and very intricate 3-dimensional structures. It could be one-dimensional, 2D, 3D, 4D. Okay, it depends on the complexity of that protein structure. But even so, if we will unfold and stretch out, okay, from the N terminus to the C terminus of that protein, they could have a one-dimensional structure. It could be linear, okay, which is showing a specific sequence of your amino acids. And very importantly, another person in the name of Charles Janovsky, an American geneticist, okay, in the faculty of Stanford University, gave us an idea of his one gene, one enzyme hypothesis, and he discovered that attenuation. So this attenuation is a riboswitch mechanism, okay, in which messenger RNA changes its shape in response to a very small molecule, and thus it binds, okay, and alters its ability for a regulatory region of a gene or an operon. So this is Charles Janovsky, the picture on your right. In 1960s, Janovsky was the first one to compare maps of mutations within a gene to a particular amino acid substitutions that resulted. And he found out that there is a relationship between the nucleotides that are found in your mRNA or DNA and the amino acids, he started working with a large number of tryptophan 2 auxotrophic mutants in your Escherichia coli. Okay, and these large number of tryptophan 2 mutants have carried mutations in your tryptophan A gene for a subunit for this enzyme tryptophan synthetase. He then made a fine structure recombinatorial map of these mutations, which is analogous or similar to the Benzer's fine structure map for the R2 region of your bacteriophage T4. With this, Janovsky was able to purify and determine the amino acid sequence of your mutant tryptophan synthetase subunits. And he found out that there is collinearity of genes and proteins. So, in the next photo or next diagram, it will show you that mutations in the gene are collinear. What happens on this level will also happen on the same spot in a very different form of your sequence of your amino acids inside the polypeptide. So you will see here in this photo that there is a relationship between the genetic map of Escherichia coli's tryptophan A gene, okay, and the positions of amino acid substitutions in mutant tryptophan synthetase proteins. And codons must include 2 or more base pairs. Okay, so you will see in the diagram that when 2 mutant strains with different amino acids, okay, at the same positions were crossed, the recombinant, okay, will produce your wild type. In this diagram, you will see collinearity of genes and proteins, and in B, the recombination within a codon. In this first image, okay, the first linear strand in blue is the genetic map for the tryptophan A mutation. And we will show here the red strand and its N terminus and its C terminus and different positions of an altered amino acid in your tryptophan A polypeptide. And in this part, we will see the wild type polypeptides and their corresponding amino acids. So the position 15 will give us lysine, the 49th position will give us glutamic acid, the 211 and 234 will give us your glycine. And in this mutant polypeptide, it will be giving us a different phenotype. Lysine will give us a stop codon, glutamic acid will give us 3. This is the valine, okay, glutamine, and the methionine. And this position in 211 and 234 will give us arginine, glutamic acid, cysteine, and aspartame. So how do researchers found out that if you combine these two or recombine these two codons, they will give us the wild type recombinants? So they tried to recombine these two mutants, 23 and 46 for the 211 position, and the 78 and 58 for the 234 position. And they found out when they combine these two mutants, they will be getting the codon, okay, for the wild type recombinant. So this one is from the first recombination, glycine, and the second one also is for glycine. So they have found out that the evidence is composed, codons are composed of more than one nucleotide. Also, Janovsky has discovered that there are different point mutations, okay, changes in only one nucleotide pair, and it will also affect the same amino acids. In both cases, Janovsky also found that recombination could occur between 2 mutations that changed the identity of the same amino acid, and recombination will produce a wild type tryptophan synthetase gene. And also, in figure 8.4a, it will illustrate that each of the point mutation in your tryptophan synthetase gene that is being characterized by Janovsky alters only a identity of one amino acid or a single amino acid. This is also true for different point mutations which are examined in many other genes, such as your human genes for rhodopsin, and the hemoglobin, the globins. So because point mutations, okay, will change only a single nucleotide pair, and most point mutations only affect only a single amino acid. Since it's collinear, okay, changes in nucleotide will change on a part of a nucleotide will also change a part of your polypeptide. Okay, each nucleotide in a gene must influence the identity of your single amino acids. So we compare the recombination maps of mutations with the amino acid sequences of mutant polypeptides, and they have established collinearity. So collinearity tells us that the order of nucleotides in the gene will correspond to the order of amino acids in your polypeptides. And there are further analysis that will demonstrate that a single codon must contain more than one nucleotide. Okay? And then each nucleotide in the gene helps encode only a certain specific single amino acids. Next would be the non-overlapping triplet codons, okay, which are set in a reading frame. In 1955, Crick and Brenner obtained a convincing evidence of the triplet nature of your genetic code, and they studied the mutations in the bacteriophage T4R2B gene. Bacteriophage are virions that affects bacteria. Okay, they have induced mutations with proflavin, which is an intercalating mutagen, and they insert itself. Okay, proflavin will insert itself between the paired bases stacked in the center of your DNA molecule. Okay, in this diagram, we will see the mutation, okay, or the mutagen proflavin that could insert itself between the two base pairs. This is your proflavin, okay, and its chemical structure. It could insert itself between the stacked base pairs within the double-stranded DNA. And researchers, okay, Brenner and Crick, exposed the strand, okay, to proflavin. The R2B plus wild type strand were exposed to proflavin, and then the mutation FC0 has popped up, okay, and they also tried to re-expose it to proflavin, and the R2B plus revertans will give us the original mutation, which is FC0, and another mutation, the second mutation, which is FC7. So in this third image, you will see the R2B plus revertans, okay, and the X wild type, okay, will yield the R2B negative recombinants. So combining, okay, recombining the original mutation and the second mutation, they will be giving us, okay, the R2B negative recombinants, which is the FC0, the first mutation, and the FC7, which is the second mutation. And in this table on your right, we have different sets of mutations that will either generate, okay, a mutant or a normal phenotypes. So this proflavin-induced mutations will give us a plus sign, which tells us an insertion, and a negative sign, which shows us the deletion. So we have, if it's only one insertion and deletion, it will give us a mutant. Double insertion and double deletion, also mutant phenotype. 4 times and 5 times deletion, or 4 times or 5 times insertion will give us a mutant. Okay, this negative and deletion and insertion, okay, will give us a wild type. Triple deletion and 6 times deletion, or the other way around, the insertion will give us a wild type. So these are some of the studies that are of what we call as the frame shift mutations in your bacteriophage T4R2B gene, shows that the codons are consisting of 3 nucleotides. Crick and Brenner, okay, began their experimentations again with the proflavin-induced R2B negative mutations, which we call as the first mutation, which is the FCO. Then they treated this mutant strain with proflavin to isolate the R2B plus revertant, and recombining this revertant with your wild type phenotype, bacteriophage, the T4, they were able to show that the revertance chromosome actually contained two different R2B plus. Codons are consisting of three nucleotides which are read in a defined reading frame. So these codons are consisting again of three nucleotides which are defined in a reading frame. And the phenotypic effects of your proflavin-induced frame shift mutations will whether depend on the reading frame which is being restored, okay, and whether the part of the gene with an altered reading frame will specify an essential or non-essential region, okay, of your polypeptide. So there are some terms that we use in this portion of our presentation. The first one is the intragenic suppressions, okay, when there are two mutations of opposite sign. It's either single base insertion and, okay, single base deletion. So this cytosine here will enter this strand, and then the adenine here will be pulled out of this strand. So they will be producing an incorrect triplet. These portions, these codons here, first, second, third, and fourth. Okay, that's intragenic suppression. The intragenic suppression that has three mutations of the same sign. It could either be three single base deletions, okay, this base pairs, the thymine, cytosine, and adenine, and they will produce this incorrect triplet here. And it could either be single base insertions. Addition of guanine, thymine, and cytosine will also give us a four incorrect triplets. Some frame shift mutations, okay, it could be single base deletion, remove one base, okay, it will produce a wrong triplet, incorrect triplet. Add one base, the guanine, it will also produce an incorrect. Using that idea, okay, how do geneticists use synthetic mRNAs to limit the coding possibilities? So if you will take a look at the previous slides, you will see that codons, okay, 1, 2, or 3, 4 codons will specify a single amino acid. So scientists were able to limit the coding possibilities, and they've used this synthetic or laboratory-made mRNAs. So the first one, the polyuracil mRNA, will generate a polyphenylalanine polypeptide. Next will be the poly-di, poly-tri, and polytetranucleotides will encode simple polypeptides, and some synthetic mRNAs such as the poly-GUAA contains stop codons in all three reading frames and thus specify the construction only of your short polypeptides. This one is poly-U mRNA, synthetic mRNA. Then in vitro translational system will add radioactive amino acids and thus producing, okay, a polyphenylalanine. This is the N terminus and the C terminus. So, okay, we analyze radioactive polypeptides which are being synthesized in this using your synthetic mRNA. And the coding possibilities using your synthetic mRNA, okay, and the created polypeptides are the ones shown in this table. Poly-U, poly-C, poly-A, poly-G, and the polypeptides produced. The repeating dinucleotides UC, okay, it will repeat UC, UC, and so on. AG, AG, and so on. UG, and so on. AC, and so on. Your trinucleotides will also produce the same way. The triplets, UUC, again, produce phenylalanine, AAG, UUG, UAC, and so on. And lastly, we have the repeating trinucleotides, thus producing this series of 4 bases, okay, of 4 amino acids in their polypeptides. So they were able to get the genetic code using the RNAs made in the laboratory. Okay, another experimentation were done by Nirenberg and Leather, and they've added trinucleotides of known sequence, okay, in combination with the tRNAs charged with radioactive amino acids to an in vitro extract containing ribosomes. So, if your trinucleotide specified the radioactive amino acid, the amino acid bearing tRNA formed a complex with the ribosomes that could be trapped on a filter. Okay, so this experiment shown in the next slide will indicate, okay, or will show that the codon CUC specifies leucine and not serine. So this is your test tube added with the serine, okay, this is the tRNA, okay, and the synthetic trinucleotide, the CUC. So once it will be poured through the filter, okay, the ribosome will indicate no radioactivity which is trapped in the filter, thus giving us the serine and this synthetic trinucleotide. On the other hand, leucine, okay, attached to the tRNA and your synthetic trinucleotide, the CUC, when placed through a filter using this attached to this ribosome, then eventually it will be read, okay, using the anticodons in this filter. Thus, it specifies your leucine, your CUC. As indicated on the first few slides, okay, there is a correlation of polarities or the directionalities in your DNA, mRNA, and polypeptide. So the template strand of DNA is complementary to both the RNA, like DNA strand, and the mRNA. And the 5' to 3' direction in an mRNA will also correspond to the N-terminus to C-terminus direction in your amino acid chain. So this one is anti-parallel, 5' to 3', 3' to 5'. Then opposite strand, 5' to 3'. Thus, the 5' will indicate the N-terminus, and the 3' will indicate your C-terminus. So this is the summary of your genetic code. It's a complete, unabridged dictionary equating the 4 language of the nucleic acids with the 20-letter language of the amino acids or the proteins. And these are the main features of your genetic code. They are a triplet, and they show the code shows the 5' to 3' sequence of the 3 nucleotides in each of your mRNA codon. The first nucleotide is depicted as the 5' end of your codon. The codons are also non-overlapping, okay? In this example, mRNA sequence 5', GAA, GUU, GAA, 3'. Okay, the first nucleotides GAA form only one codon, the next will be forming the second, and so on. Okay, each nucleotide is part of only one codon. So that's the nature of your genetic code. There are also codes that signify stop codons or termination codons. We have the UWA, UWAG, UGA, and they do not encode a certain amino acid, thus terminating translation. And the code is said to be degenerate, or the degeneracy of the code. It means that more than one codon will specify a single amino acid, and the code is nevertheless unambiguous because each codon will specify of only one amino acid. And the machinery of your cell will scan through mRNA, okay, using your polymerase, RNA polymerase, from a fixed starting point, which then establishes the reading frame. So as we see later in our future lessons, the nucleotide AUG will signify the start codon or the initiation codon, or this AUG is the methionine, and that's the marker of your mRNA, okay, where the certain polypeptide will begin to be translated. Next will be the corresponding polarities or the directionalities from moving from the 5' to 3' end of the mRNA. They are interpreted sequentially, okay, starting from the N-terminus of your amino acid moving toward the C-terminus of your polypeptide. And mutations are also modifying the message that are encoding in a sequence of nucleotides in a very subtle ways. We have frame shift mutations, okay, they are nucleotide insertions or deletions that alter the genetic instructions for the polypeptide constructions by each of the reading frame. It could be missense mutations. It will change a codon, okay, for one amino acid to a codon for a different amino acid. It could be nonsense mutations and changing a codon for amino acid to UWAG, UWA, UGA, or the termination codons. So there is a question, okay? What are the effects of mutations on polypeptides that are helping us to verify the code? Because the experiments that were used prior to this slide are all in vitro studies, okay? They are using cell-free extracts and synthetic mRNAs. So the logical question thus arose: do living cells construct polypeptides according to the same rules? Okay, we will compare it to the ones which are living. We're comparing it to the ones which are actually happening in the real world. So there is an experimental verification of the genetic code, okay, using your single base substitutions that are explaining the amino acid substitutions of your tryptophan 2 mutations and your tryptophan 1 revertants. So it will indicate also here that the genetic code predicts the amino acid alterations in yellow, and they will arise from single base pair deletions and suppressing insertions. So this is your position and your polypeptide 211, okay? The amino acid in the wild type, we have the glycine or the GGA, and the possible mutations of the mutant polypeptides are the arginine and the glutamic acid, and these are the codons, AGA and GAA. So there are different reversions changing, okay? One of the three triplets, okay, or parts of the triplet. So we have AUA, ACA, AGC or AGU, and GGA. So we have 4 reversions, okay, from arginine, and also 3 reversions from glutamic acid. And this wild type mRNA in the polypeptide has underwent, okay, insertion and deletion. So there is an addition of guanine and deletion of adenine, and it will show you a double mutant mRNA and polypeptides, thus giving us this incorrect, okay, series of codons. So this is accompanying intragenic suppressions. So it will also give us the idea that the genetic code is almost but not quite universal. So we will know that all of the cells that are alive today use the same genetic code. And one indication or early indication of this uniformity among organisms is that the translational system that are derived from one organism could be used, okay, the mRNA from another organism to somehow convert, okay, genetic informations to the encoded protein. Another proof to this almost but not quite universal genetic code is the study of the rabbit hemoglobin mRNA. When they are being inserted to the frog eggs or added to cell-free extracts of a wheat germ, okay, these mRNA will direct the synthesis or creation of rabbit hemoglobin proteins. And most recently, comparing the DNA and protein sequences have revealed a very perfect correspondence according to the genetic code between codons and amino acids in almost all organisms examined. Thus, the genetic code is conserved, okay? This is a very good indication that the universality of the code has evolved in the early history of life, and once it has emerged, it remained constant over billions of years. It remained stagnant, and because part of it, organisms evolve, okay, would have a little tolerance for change. There are, okay, changes, okay, or single change in the genetic code that disrupts different productions of hundreds of thousands of proteins in your cell, okay? From the DNA polymerase that is very essential for the replication to the RNA polymerase that's required for the gene expression to the tubulin proteins that compose the mitotic spindle, and such a change would therefore be lethal, and that's a fact, okay? Another thing is one of your exceptional genetic codes. The study was done, okay, because there are some or few exceptions to this universality of the code, and researchers were quite amazed with this idea. In some species of the single-celled eukaryotic protozoans, we know them as ciliates, the codons UAA and UAG, which are nonsense codons, in most organisms will specify the amino acid glutamine, and other ciliates, UGA, okay, is the third, okay, stop codons in most organisms will specify cysteine. So that's not the case. They are participating in a very different, okay, or very exceptional way of using these triplets of codons and in their systems. These ciliates are using the remaining nonsense codons as stop codons. It's very unique, right? Also, there are some exceptional genetic codes existing in the mitochondria, which is also containing the DNA of your cell. Okay, this mitochondrion is semi-autonomous. They are self-reproducing organelles within the eukaryotic cells, and very importantly, they are the sites of your ATP formation, and they have their own chromosomes and its own apparatus for gene expression. So this photo, okay, will show you a long mitochondria, okay, but however, in the mitochondria of your yeast, the CUA will specify threonine instead of leucine. It could be also an indicator that ciliates and mitochondria has tolerated a very few changes, okay, in the genetic code, because the alterations affected a very few proteins. And for instance, the nonsense codon UGA, okay, or the stop codon UGA might have found only an infrequent use in one kind of a primitive ciliate, and for it to switch to a sense codon, okay, would have made a very, very tremendous difference in protein production. And similarly, mitochondria have been surviving a very few changes in the code because they only synthesize only a handful of proteins. So there we understand why they are exceptional. So, but although cracking the code made it possible to understand the broad lines of information and flow between gene and protein, it did not explain exactly how a cell machinery, okay, accomplishes gene expression. And this is our focus as we present the details of our next video, one for transcription and another one for translation. Okay, so that's all we have for today. Thank you so much for listening, and don't forget to subscribe to my channel. Thank you, and goodbye.